Hello friends, this video on respiration in plants part 10 is brought to you by examfear.com. No more fear from exam. Let us now talk about the third step of respiration, that is the Krebs cycle. So the name is little weird. What is Krebs cycle? What is this Krebs all about? Okay, let us first try to understand why the name Krebs cycle. It is named after a scientist called Hans Krebs who demonstrated it for the first time. So here you can see the picture of Hans Krebs. So after his name, it was called as Krebs cycle. It is also known as citric acid cycle. Why is it called citric acid? Because the first stable product which is formed here is a citric acid. So citric acid is the first product which is formed. That is why it is called citric acid cycle. It is also known as tricarboxylic acid cycle or TCA cycle. Why tricarboxylic acid? Because the citric acid has 3 carboxyl groups. Tri means 3, carboxylic, that is carboxyl groups. Since citric acid has 3 carboxyl groups, that is why it is called tricarboxylic acid cycle. Now, in many of your textbooks, you would find these names being used interchangeably. Krebs cycle, citric acid cycle, and TCA or tricarboxylic acid cycle. Now, as the name says, it is a cycle. That means the starting point and the end point are going to be the same. That is why it is a cycle. It is a circular process. So, in Krebs cycle, again, it is a multi-step process. It is again not a single step process. When you talk about a multi-step, it consists of total 8 steps. Okay, so just imagine, just think of the entire process of respiration. First was glycolysis, which was a 10-step process, then 1 step for pyruvate oxidation, and now again 8 steps for Krebs cycle. So we are almost reaching some 19 steps here itself. After this, we'll have the electron transport chain. So just imagine the number of steps, how many multiple steps are there in the process of aerobic respiration. Okay. So anyways, so we will talk about each of these 8 steps in detail one by one. Now here, citric acid is the first product as well as the final reactant. So citric acid is the first product which is formed, and again it is the final reactant. How it is the final reactant? We will see that as we go through it and go through the cycle. And that is why the other names of the cycle are named after citric acid. So let us start with step 1. So let us look at the first step of the Krebs cycle. So what would be the starting material for Krebs cycle? The product of pyruvate oxidation, that is acetyl coenzyme A. So this is going to be our starting material. So what is going to happen here is condensation with oxaloacetic acid occurs. So when I say condensation, it basically means that oxaloacetic acid and acetyl coenzyme A will combine together to form a new compound. Now acetyl coenzyme A is a 2-carbon compound and oxaloacetic acid is a 4-carbon compound. Now when condensation takes place between a 2-carbon compound and a 4-carbon compound, what is expected? Right, a 6-carbon compound will be formed. So what is that 6-carbon compound? Let us quickly see. The 6-carbon compound is going to be citric acid, and the enzyme which plays a role in this condensation is citrate synthase. So the product is citric acid. This is how it will take place. Acetyl coenzyme A will combine with oxaloacetic acid to form citric acid plus coenzyme A. So basically this acetyl group will combine with this oxaloacetic acid to form citric acid, and the coenzyme A will get separated and it will separately be released. So that is what happens in step 1, and that is why we see that the first stable product which is formed in a Krebs cycle is citric acid, and that is why it is also known as citric acid cycle. Another product which is formed is coenzyme A. Let us look at step 2. Here the starting material is citric acid or citrate, whatever you call it. Isomerization occurs. That means citric acid will get converted to one of its isomers. So what kind of enzyme will be there? Some isomerase enzyme. So here that enzyme is aconitase. So this enzyme will isomerize the citric acid by removal and addition of waters. First it will remove water and again it will add water. And what will be formed? The product will be isocitrate. And then the next step, that is step 3. Here the starting material will be isocitrate or isocitric acid. Decarboxylation will occur, that is removal of carbon dioxide will take place. So here you have isocitrate, which is a 6-carbon compound, and during this process carbon dioxide will be released. So it will become a 5-carbon compound. And what is that 5-carbon compound which is formed? That is alpha ketoglutaric acid. So this is the 5-carbon compound which is formed. And also during this decarboxylation, NADH is also released. NADH is also formed. So the enzyme which plays a role here is isocitrate dehydrogenase. And the product formed is alpha ketoglutarate or alpha ketoglutaric acid and NADH. So here if you see, wherever NADH gets released, that step is important because NADH can cause ATP synthesis, and ATP synthesis is what we are looking at primarily. So NADH is important. Also CO2 gets released in the process because this decarboxylation happens by removal of CO2. Step 4, the starting material will be alpha ketoglutaric acid, and here again decarboxylation occurs. As I said, during Krebs cycle, multiple decarboxylation will occur in order to cause the complete oxidation of pyruvic acid. So after another decarboxylation with the enzyme alpha ketoglutarate dehydrogenase, the product formed is succinyl coenzyme A. So alpha ketoglutarate is a 5-carbon compound. This in presence of the enzyme alpha ketoglutarate dehydrogenase, it will lose carbon dioxide, that is decarboxylation will take place. Also during this process, NADH will be produced. Now when one carbon is removed, a 4-carbon compound is formed, and that 4-carbon compound is succinyl coenzyme A. So this is the product which is formed. Also NADH is formed and CO2 released. Step 5, starting material would be succinyl coenzyme A. So what happens here? Phosphorylation occurs. So for phosphorylation, again you need an enzyme, and the enzyme here is succinyl coenzyme A synthetase. So this is the enzyme. So what happens in this step is you start with succinyl coenzyme A, which is a 4-carbon compound. This in presence of the enzyme succinyl coenzyme A synthetase, phosphorylation will happen. So during phosphorylation, what happens? One phosphate group is removed. So one phosphate group is removed from here and it is passed on to GDP to form GTP. Now you might be wondering, what is this GDP now? Till now we were hearing only about ATP and ADP. Now what is ATP? That is adenosine triphosphate. So GTP is nothing but guanosine triphosphate. So the way in ATP you have adenine amino acid, similarly in this case you have the guanine group. So that is the only difference. So this is guanosine triphosphate, and this is guanosine diphosphate. So the one extra phosphate from here is taken up by GDP and it gets converted to GTP, and the product which is formed is succinic acid, and this succinic acid is also a 4-carbon compound. So here if you see substrate level phosphorylation take place. Now what is this GTP? This GTP can be used as an energy source in protein synthesis. GTP can be very easily converted into ATP by nucleoside diphosphokinase. So that is the enzyme. Nucleoside diphosphokinase. So this is the enzyme which can actually convert GTP into ATP. So basically production of GTP in this step is also crucial because it is causing to synthesize ATP. So the products formed are succinic acid or succinate and GTP, that is guanosine triphosphate. So now we have reached till succinic acid, which is a 4-carbon compound. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.